The specimen before me defies every limitation printed on the back of a glossy industrial fertilizer sack. We are looking at a Solanum lycopersicum, a common tomato plant, but there is nothing common about the violence of its growth. The stalk is as thick as a laborer's thumb, vibrating with turgor pressure, struggling to hold up a canopy of fruit that has tripled the yield of the control group in the neighboring plot. There are no blue crystals here, no synthetic nitrogen pellets dissolving in the mud. If you dig 3 inches beneath the surface, right into the rhizosphere, you won't find a receipt for a $50 bottle of patented growth hormone. You will find a crime scene, a black, oxidizing, slimy mass of organic decay that most of you would instinctively recoil from. It is a single rotting banana dissolving into the soil matrix. For decades, the agricultural giants have conditioned you to view this rot as waste, to seal it in plastic and ship it to a landfill while you swipe your credit card for sterile, laboratory-grade macronutrients. They sold you cleanliness, but they stole your yield. This isn't gardening. This is forensic biology. What we are witnessing is not a miracle, and it certainly isn't magic. It is a precise high-efficiency transfer of bioelectric potential and potassium ions that has been silenced by a market dependent on your ignorance. The question you need to ask isn't how this garbage grew a monster. The question is why you were taught to throw the fuel away and buy the fumes. To understand why you are currently standing in a garden center staring at a wall of plastic bottles, we must rewind the tape to 1947. The Second World War had ended, but the machinery of industrial chemistry was spinning at maximum velocity. The massive factories that had spent years synthesizing ammonium nitrate for explosives suddenly found themselves without a war to fight, yet they possessed a terrifying surplus of production capacity. They needed a new enemy, and they found one in the soil itself. A decision was made in boardrooms far removed from any farmland to rebrand these surplus munitions as agricultural salvation. This was the dawn of the NPK era. Nitrogen, phosphorus, potassium, stripped of their biological context and sold as white crystalline salvation. Before this calculated pivot, agriculture operated on the law of return. It was a closed loop where nothing was wasted, and waste was merely energy waiting to be unlocked. Farmers and localized growers understood that the harvest took nutrients from the earth, and the only way to sustain the system was to return organic mass back into the furrow. The banana peel, rich in the very potassium the fruit had extracted, was not garbage. It was a battery. It was a packet of solar energy and mineral density that belonged back in the root zone. But you cannot patent a rotting fruit. You cannot put a barcode on a natural cycle of decay, and you certainly cannot build a global supply chain on a resource that every household generates for free. The erasure was systematic and psychological. Through aggressive marketing campaigns in the 1950s and 60s, the chemical conglomerates waged a war on primitive methods. They painted the natural biological processes of fermentation and decomposition as dirty, unpredictable, and unscientific. They replaced the dark living complexity of humus with the sterile certainty of the chemical bag. They taught a generation of growers that soil was nothing more than an inert medium, a sponge whose only purpose was to hold up the plant while the chemical company provided the life. The knowledge of the potassium cycle, the understanding of how organic matter buffers pH and feeds the microbiome, was not just forgotten, it was actively ridiculed. They severed the hand that fed the soil, creating a dependency model that required you to buy back the very fertility you were throwing into the trash bin. We need to strip away the nursery rhymes about feeding plants and start talking about cellular hydraulics. If you think your tomato plant is just passively sitting there waiting for food, you are missing the violent reality of its internal engineering. The plant is a pressurized system, and potassium, the element designated as K on the periodic table, is the master of that pressure. It is not a structural building block like calcium. It is a mobile functional agent, a bioelectric spark that travels through the plant's vascular system like a courier with a high-priority clearance. When we analyze the rotting banana, we aren't looking at mulch. We are looking at a dense cache of K+ ions waiting to be deployed into the plant's potassium pump. This mechanism is purely physical. At the microscopic level, your tomato leaves are covered in thousands of tiny mouths called stomata. These gates control the intake of carbon dioxide, the breath of life, and the release of water vapor. The plant cannot muscle these gates open. It uses the potassium pump. When K+ ions flood into the guard cells surrounding the stomata, water follows them instantly through osmosis, swelling the cells until they bow outward and snap the gate open. Without adequate potassium, the gates remain shut, the plant suffocates, and photosynthesis stalls. But here is the critical divergence between the industrial bag and the biological reality. The source of the ion dictates the reaction of the root. The synthetic option, potassium chloride, is mined from the earth and refined into a harsh salt. When you dump this pink or white crystal onto your soil, you are bombarding the delicate root hairs with a saline shock, forcing the plant to expend precious energy just to filter out the chloride toxicity while scavenging for the potassium. It is an act of chemical violence. The banana, however, holds potassium in a bioavailable organic matrix, often chelated with carbon chains. When it breaks down, it releases K+ ions in a rhythm that matches the plant's natural uptake capabilities. It activates over 60 distinct enzymes responsible for protein synthesis and fruit density without burning the microscopic bridges that allow the root to function. This is not about being natural. It is about thermodynamic efficiency. We must now address the invisible volatile agent that terrified the commercial shipping industry so much they spent millions inventing ventilation systems to scrub it from the air: ethylene. In the sanitized world of grocery logistics, this hydrocarbon gas is the enemy, a ripening accelerator that turns profit into mush during transit. But when you bury that blackening banana 6 inches deep, you are not destroying a commodity. You are trapping a powerful hormonal signal directly in the root zone. You are essentially wiring the tomato plant's dashboard to a new frequency. This is where the physics of soil porosity becomes critical. In the open air, ethylene dissipates instantly. Underground, trapped within the lattice of clay and silica, the gas creates a localized atmosphere of urgency. When the root tips of your tomato plant detect this concentration of C2H4, they do not perceive it as food. They perceive it as a command code. The gas binds to specific protein receptors on the root membrane, triggering a signal transduction pathway that overrides the plant's default leisure mode. It screams biological timeline acceleration. The plant interprets this ethylene presence as a cue that the environment is evolving, perhaps even decaying, and in a stroke of evolutionary genius, it responds not by dying, but by shifting its metabolic priority from vegetative safety to reproductive desperation. It stops lazily putting out green leaves and starts diverting massive amounts of sugar and energy into flower production. This is the ethylene trigger. You are hacking the plant's survival instinct, using the dying breath of the banana to fool the tomato into believing it must reproduce, create fruit, immediately and abundantly. It is a hormonal sleight of hand that no synthetic nitrogen pellet can emulate, because nitrogen only fuels size, while ethylene dictates purpose. You might imagine the tomato roots gnawing on the buried fruit like a dog on a bone, but that is a cartoon simplification that insults the complexity of the rhizosphere. The plant does not eat the banana. The plant lacks the digestive enzymes to break down complex organic matter. Instead, it outsources this labor to a mercenary army that has been starving in your soil, waiting for a payload of carbohydrates. When you bury that sugar-dense banana, you are not feeding the crop. You are ringing the dinner bell for the dormant microbial workforce. Within hours of the peel rupturing, the population of native bacillus bacteria and soil yeasts goes exponential. They do not just consume the fructose and sucrose leaking into the dirt, they frenzy. This is the sugar microbe bridge. These microscopic laborers, billions of them in a single handful of earth, swarm the carbon structure of the fruit. As they metabolize the sugars, they perform the critical alchemy of mineralization. They strip the bound potassium, calcium, and phosphorus from the solid organic matter and excrete them as dissolved ions. This process transforms inert waste into a bioelectric current that the plant can actually conduct. The microbes are effectively the external stomach of the tomato. They digest the raw fuel and release organic acids that chelate the minerals, wrapping them in a chemical embrace that makes them instantly absorbable by the root hairs. Without this microbial bridge, the banana is just garbage. With it, the soil becomes a digestive engine, processing nutrients with a catalytic efficiency that no synthetic salt, which kills these very same microbes on contact, can ever hope to replicate. Here lies the grand architectural flaw of modern agriculture, a deception built on the illusion of speed. The industry sells you bags of fast-acting synthetic nutrients, promising overnight vigor. What they are actually selling you is a high-salinity time bomb. When you dissolve those blue or pink crystals, essentially concentrated mineral salts, into your watering can, you are not creating a nutrient broth. You are spiking the electrical conductivity of the soil to dangerous levels. In physics, this creates a catastrophic reversal of osmotic pressure. Instead of water flowing passively into the roots, the high salt concentration in the soil pulls water out of the plant. This is the mechanism of fertilizer burn. It is not a burn at all. It is cellular hemorrhage. The plant dehydrates from the roots up, gasping in a sea of plenty. Compare this to the banana. The decaying fruit does not dissolve. It disintegrates. It creates a colloidal humus, a sponge-like buffer that holds moisture and nutrients in a suspended state. There is no osmotic shock because there's no sudden spike in salinity. The potassium is released molecule by molecule, governed by the metabolic rate of the microbes, not the solubility of a factory salt. But the deception goes deeper. The synthetic nitrogen forces the tomato to grow cells that are large, watery, and thin-walled, structurally weak giants. These distended cells are unable to produce the complex secondary metabolites that act as natural immune systems. They become beacons for aphids and blight. And here is the genius of the business model. Once your artificially pumped-up plants inevitably succumb to pests because their cell walls are paper-thin, who do you turn to? The same company holding a bottle of pesticide. They break the plant's legs with one product and sell you the crutches with another. Amateurs bury the fruit whole and wait for nature to file the paperwork. We do not have time for the slow bureaucracy of natural decay. To weaponize the potassium, you must first destroy the structural integrity of the delivery system. The peel is a cellulose fortress designed to keep bacteria out. So your first move is mechanical demolition. You will take that blackening banana and slice it, flesh, peel, and stem, into pieces no larger than a coin. We are maximizing the surface area to volume ratio, exposing the inner carbohydrate matrix to the microbial swarm. But do not bury it yet. You need to prime the charge. Place this slurry into a sealed jar with tepid water and a teaspoon of raw sugar or molasses. Shake it. You are creating a temporary anaerobic reactor. For 3 days, you let the pressure build in the dark. You are cultivating a localized explosion of lactobacillus bacteria that will begin enzymatic hydrolysis before the matter even touches the dirt. When you finally unscrew that lid and hear the hiss of escaping gas, you know you aren't holding garbage anymore. You are holding a pre-digested potassium grenade ready to detonate. Precision is the difference between a fertilizer and a poison. If you dump this fermented potassium charge directly onto the main stem, you are inviting disaster. The collar of the plant, where the stalk meets the soil, is the most vulnerable point for fungal ingress. You will rot the structural highway before the fuel ever reaches the engine. Instead, you must locate the drip line. Visualize the canopy of the plant. The feeder roots extend laterally to match the reach of the leaves. This is your injection zone. Measured exactly 6 to 8 inches from the central stalk, dig a trench 4 inches deep. This depth is non-negotiable. Too shallow and UV radiation sterilizes your microbial workforce. Too deep and you hit the anaerobic layer where oxygen-starved bacteria produce toxic sulfides instead of available ions. You want the aerobic sweet spot. Pour your fermented slurry into this trench. You are creating a subterranean depot, a localized concentration of high-voltage nutrition that forces the plant's lateral roots to aggressively branch out. Within 48 hours, the root hairs, sensing the chemical gradient of the leaching potassium, will physically turn and surge toward this pocket. You aren't just feeding the plant, you are steering its underground architecture. Fast forward 30 days. The forensic evidence is now undeniable. You're not looking at the same plant that struggled in the control group. The main stalk has thickened to the diameter of a garden hose, vibrating with turgidity, its cell walls reinforced by the silica and potassium matrix you injected. The leaves are not merely green, they are a deep, almost black emerald, a visual indicator that the chlorophyll density has maxed out the plant's photosynthetic capacity. But the real shockwave is the canopy. It hasn't just grown, it has erupted. The flower density is not a percentage increase, it is a multiplication event. Where the chemical fertilizer produced a few polite clusters, the banana-fed root zone has triggered a violent detonation of yellow inflorescence. This is the direct result of the potassium pump operating at full capacity, moving sugars from the leaves to the fruit sites with zero resistance. The fruit set rate, the ratio of flowers that actually become tomatoes, is nearly perfect. There is no blossom drop here because the plant is not under stress, it is under command. And then there is the taste, a metric the industry tries desperately to ignore. If you refract light through the juice of a supermarket tomato, you get a brick score, a measure of dissolved sugars, of maybe 4. It is red water. Your specimen, fed on the complex decay of the banana, is pushing a Brix of 12. The fruit is heavy, dense, and complex because it wasn't pumped up with saline water. It was built with mineralized biology. You haven't just grown a tomato, you have embarrassed the supply chain. This was never really about tomatoes. It is about the shackles of convenience. Every time you throw a banana peel into the trash and drive to the store to buy a plastic bottle of blue liquid, you are signing a contract of dependency. You are agreeing that the chemical industry understands nature better than nature itself. You are trading your autonomy for a barcode. The system is designed to make you feel helpless, to convince you that fertility is a product you must purchase rather than a cycle you can command. But the moment you stop viewing that black, oozing fruit as waste and start seeing it as energy, you cease to be a consumer. You become an alchemist. You are transmuting what society calls garbage into biological gold. The science is not hidden, it is just ignored. The physics of the potassium pump, the ethylene trigger, the microbial bridge, they are all waiting for you to engage them. So here's the final question, the only one that matters. Are you going to keep paying for the illusion of growth, or are you ready to build the real thing? The revolution does not begin in the streets. It begins 6 inches underground.